This protocol presented the method to transiently improve cardiac function in Duchenne's muscular dystrophy mice by transplanting exon derived from normal myogenic progenitor cells.
The exosome transplantation can improve heart function in DMD mice in association with increased expression of dystrophin in recipient hearts.
Appropriate method of exon isolation, purification, and intramyocardial transplantation contribute to the success of the exon transplantation to improve cardiac function for mice with Duchenne's muscular dystrophy.
MPC-derived exosome therapy might improve skeletal muscle function by exosome-mediated normal dystrophin mRNA transfer. This video demonstrates the critical techniques for exosome purification, intramyocardial exosome delivery, and echocardiography post-transplantation.
The demonstration of echocardiography will be done by Shen, technician from my lab.
To begin this procedure, seed 5 million C2C12 cells into a 15-centimeter culture dish with 20 milliliters of complete DMEM containing 10% FBS and antibiotics. Incubate at 37 degrees Celsius with 5% carbon dioxide. Next, use a swinging bucket rotor to ultracentrifuge FBS at 100,000 times G and at 4 degrees Celsius for 18 hours to prepare exosome-depleted medium. Discard the pellet and collect the supernatant.